Hello everyone, I'm Shilu from University of Wisconsin-Madison. Today I will talk about defining cell type varying networks of chromatin marks and transcription factors underlying cell fate transition dynamics. A regulatory network is a set of macromolecules consists of proteins such as transcription factors, signaling proteins, RNAs, and chromatin marks that interact to control the level of expression of various genes in a given genome. At a high level, we are interested in understanding regulatory network dynamics on a lineage. Several papers have measured cell type-specific data to study the dynamic regulatory networks during reprogramming or differentiation. The challenge, however, is that regulation of gene expression is complex, especially in higher eukaryotes. In particular, there are two key challenges. We need methods to capture the relationship between chromatin marks, TFs, and expression levels in one cell type, and understand how changes in chromatin state and TF binding influence changes in expression levels. To overcome these limitations, we present cell type varying networks, a probabilistic graphical model-based approach that uses multitask learning to capture the relationship between chromatin marks, TFs, and expression levels in each cell type and models the relatedness by a structure pair. This enables us to understand how changes in chromatin state and TF binding influence changes in expression levels. The input of CVN is a lineage tree and cell type-specific data of chromatin marks, accessibility, expression, and TF binding. The output is a set of cell type-specific networks for each cell type. We compared CVN to two approaches, ChromNet, which learns a single network for all cell types, and InDepth, which learns a network per cell type, but does not share information across cell types. We simulated four networks from a lineage tree for four cell types and generated simulated data from four graphical Gaussian models. On simulated data, CVN networks are able to recover the interactions better than ChromNet and InDepth with a higher AUPR. Examining the precision recall curves, we observed that CVN has higher precision and recall compared to ChromNet and InDepth. This suggests that CVN is able to predict the true interactions accurately. Next, we apply CVN to published ChIP-seq, ATAC-seq, and RNA-seq datasets for four stages during mouse reprogramming to infer the cell type-specific networks of chromatin marks, TF, and expression at each stage. To gain insight into the differential interactions across these four stages during the reprogramming process, we subsampled the data 50 times and applied CVN in the stability selection mode to estimate the confidence score for each age in the inferred networks. Then we did k-means clustering on this age-by-cell matrix to find different patterns across cell stages. We choose 13 clusters, which several of these subnetworks exhibit different patterns of conservation. Let's dig into the subnetworks in cluster 9, which is pre-APSC and ESC specific. We found a large number of cell type-specific interactions in pre-APSC and ESC, but were absent in MEF and MEF48. Here is a zoom-in of the subnetworks. Key chromatin marks like H3K9 acetylation, H3K27 acetylation are interacting with different TFs such as SOX2 and ZFX and affecting gene expression. SOX2 is a pluripotency marker critical for controlling the expression of genes in embryonic development. ZFX has been shown to control the self-renewal of embryonic stem cells. In summary, CVN is a powerful framework to infer cell type-specific interactions between chromatin marks, TFs, and expression. And CVN is able to predict the true interactions accurately and capture both the cell type-specific interactions and conserved interactions across cell lineage. Application of CVN on data during reprogramming identified subnetworks and chromatin hubs that are common and differentially varied across the cellular stages and provided insight into network changes during reprogramming. I would like to thank Susimita and everyone from the Roy Lab and our finding resources. Thank you.